Hello everyone, we're Group 122, and our project is on EEG-to-Text for speech-impaired individuals using CNN architecture. I'm Ishita, and we have Arman and Pierce in our group as well. So let's look at the motivation. Imagine losing your ability to speak due to an injury or illness. Brain-computer interfaces aim to restore communication by decoding speech directly from brain activity. Today, we will be presenting you our baseline model, talking about our architecture and the final evaluation metrics to get the accuracy and scores for our project. So let's look at our data set. Our data set was a Dutch study where we had 10 participants, 10 native Dutch participants with pharmacoresistant epilepsy, with 5 female and 5 male, and they were shown 100 random Dutch words, and they had EEG electrodes connected to their brain, and while they saw the words, they also read those words through a microphone. So there were two input data collected. Here's a small visualization, and let's move on to what exploratory data analysis we conducted. So for the EEG data, the EEG time series data was detrended. It was passed through a bandpass filter for 70 Hz to 170 Hz, which is the peak frequency range for speech production. Think of this like tracking the volume of the brain activity over time. On the other hand, for the audio data, we broke it down into time windows from 50 millisecond length with 10 millisecond slide. Further, with the EEG data, we converted it through a Hilbert envelope, which took the maximum amplitude. And this Hilbert envelope, we computed the mean value of the Hilbert envelope for every window of 50 milliseconds. On the other hand, for the audio data, we computed a short-term Fourier transform for each time window, then stacked these time windows into a spectrogram, which is amplitude of frequency over time. Further, we took the MEL spectrogram, which is frequencies in the MEL spectrogram are MEL scale, whereas the log of the amplitude is in decibels. Let's move on to our baseline model, which is personalized for each participant.
Yeah, so as you mentioned, this was done for each individual participant. So we have a regression model for each one, all 10 participants. The input for the model was the array of the means for the array of means of the Hilbert envelope for each time window and for all electrodes that were sampled. And the ground truth was the log MEL spectrogram. Both of these pieces of data had a train test split taken of them. For the entire data set of the EEG, which we'll call the EEG feature matrix, a PCA analysis was performed. The first 50 principal components were extracted and put into a smaller matrix known as the PCA matrix. The EEG data was then projected onto this PCA matrix as a form of data reduction to consolidate the information provided by the data. Then the training version of the PCA projected EEG features were used to train a linear regression model where there was a learned weight matrix. The input, as I mentioned, was the PCA EEG features, and then the ground truth was the training split of the log MEL spectrogram. From there, the learned weight matrix was used to help predict where, again, the input was the test split of the PCA projected EEG features, and which produced a predicted spectrogram. This predicted spectrogram was then compared to the test split of the log MEL spectrogram, which again was the ground truth. From there, we could compute metrics. And then Armand is going to talk about the architecture.
All right, so the architecture that we opted for is a 2D CNN. We chose a CNN as opposed to other architectures due to our limited data. The input to the CNN is the EEG signal split by brain region and then split in time by word. So basically, a single data point represents the EEG data across brain regions for a single word. So this data was sent into our 5 layers CNN architecture, and it outputs a audio MEL spectrogram, which is split into the 23 frequencies that are key to human speech. So for our evaluation metrics, we decided to use mean squared error as a metric, which is just a standard metric for a regression problem. Compared to the baseline model, we actually did a little bit worse, but keep in mind that our model was generalized across all participants, and the baseline model was specialized for each participant. Given that we had pretty limited data, we only had 10 participants. This is actually pretty good, and if we were given a larger data set, this model could definitely be expanded, and it would definitely be possible to get better results. So thank you for listening. Here are our references.
Thank you.